please do it. Do it because, it changed completely my career. And I think it's a great opportunity, but not just for the funding itself, that obviously is what we need to move forward in our career, for sure. But also for the visibility and because the prestigious of these grants is very well known in the community of hematologists.
Welcome to EHA Unplugged, the official podcast channels of the European Hematology Association, EHA. I am your host, Isabel Olivera, and today we have the pleasure to have with us Dr. Ana Río-Machín. Welcome.
Thank you very much for having me.
So, Dr. Ana Río-Machín is a PI at the Hematology Experimental Lab Autónoma de Madrid, and she's also an honorary lecturer at the Center for Hemato-Oncology at the Barts Cancer Institute in the Queen Mary University of London. Her research aims to improve the understanding and management of familial AML, acute myeloid leukemia, and focuses on exploring germline mutations in families with AML. Dr. Río-Machín's work was supported by an EHA Junior Research Grant. Her results have been recently published, and we will be discussing them today. So we will start by analyzing why she's studying familial AML, then why she has focused on a certain translocation and what the consequences of this translocation are and how they activate key leukemia genes. And finally, we will see how this is clinically relevant at the moment. So let's start by the basis. Looking at the big picture, why work in familial AML?
Thank you. There are two big groups of acute myeloid leukemia, the sporadic cases, but we are always forgetting about the familial cases that are the ones where we are inherited a mutation that can cause the disease. So this was recognized very recently by the WHO classification of myeloid neoplasms. So before 2015, even if we knew that there were some aggregation in families, nothing was really done for the diagnostic of these families. We were investigating in new genes that could be like causing this predisposition to acute myeloid leukemia and other myeloid neoplasms. So we considered that it was really, really relevant to look into these families, identify the genetic cause of these, especially at the point of the transplantation, because so far we know that the cure of these patients is the transplantation. The best donor is normally a familiar or a relative of these patients. But if the patient, if the relative has also the germline variant, the recipient will develop the disease again. So this is what we call donor-derived leukemia, and it's really aggressive. So we need to avoid that. So we need to understand the genetics behind familial cases. But then my work also focuses on the sporadic disease. So I have two main lines of research of my research. One focuses on familial cases, but the other is in a sporadic disease. Like, for example, this rare subtype that is the patients with the chromosomal translocation between chromosome 6 and chromosome 9.
So this translocation between chromosome 6 and 9, you say this is sporadic, it's not familial.
It's not familial.
So this creates a fusion protein. What are the consequences of this fusion protein? What does it do?
That was exactly what we wanted to understand with this project, because it is a recognized subtype, is again in the myeloid classification by WHO. But so everybody knows that it exists, it's quite frequent, but the consequences of and the function of this fusion gene, it was still unknown, and the patients didn't have any specific therapy for. So what we did know is the translocation between chromosome 6 and chromosome 9 results in a fusion gene between the DEC gene and NUP214 gene. So the consequence of this fusion gene is a chimeric protein, half of DEC and half of NUP214. So that was known, but how this chimeric protein induces the development of acute myeloid leukemia was still unknown, and the patients these patients with this translocation have a really poor prognosis, especially when this translocation occur at the same time with the FLT3-ITD mutation that is quite frequent. Normally, in more than 70% of these patients also appear this FLT3-ITD mutation. So the prognosis of these patients is really poor. The age of diagnosis of these patients is younger than the average for acute myeloid leukemia. Or they normally have a really high rate of relapse, because there is no treatment. So they respond, but then they relapse because the clone with the translocation is still there, and it causes the relapse of the disease. So we really wanted to understand how this chimeric protein could induce leukemia, and if there was something that we could target to kill these leukemic cells.
So what are the targets of this fusion protein?
So the main finding of our project, and that we are really proud of, is we found that DECNUB-214 acts as a transcription factor. So it binds to the regulatory region of key leukemia genes, like Hox genes of FOXY1, and this induces the overexpression of these key leukemia target genes. So then what we know is the overexpression of FOXY1 or Hox genes induces leukemia. So this is already known because they are genes overexpressed in other subtypes of leukemia. But what we identified was what was causing this overexpression. That was the binding of DECNUB-214 to the regulatory regions. So then understanding why these genes are inducing leukemia, it was quite well studied before, because FOXY1, for example, blocks differentiation, but is one of the hits that normally occur to induce leukemia development, and also as well the overexpression of Hox genes that are really, really well-known targets and are overexpressed in other subtypes like the ones with KMT2A rearrangements. So the novelty was identifying this new role for DECNUB-214, inducing the overexpression of these genes. And what it was more important, it was the DECNUB-214 binds to this region with the help of XPO1. And XPO1 is a protein that binds to DECNUB-214, and it's required this binding for the overexpression of Hox genes and FOXY1, among other genes. And it was really interesting because we had a good representation of patients with the 6-9 translocation, given that it was a very rare entity. And we compare the response of these patients, the cells of these patients with the T6-9 translocation, with the response for other AML subtypes. So we treating all these cells with more than 527 compounds. And it was really interesting to find that on the top of the list, so the compounds that were more specific and more selective for the T6-9 patients were the XPO1 inhibitors. So there was a clear relationship between our finding, so identifying XPO1 being super specific and efficient for the T6-9 patients compared to other AML subtypes, and the fact that DECNUB-214 interacts with XPO1. So then we went a step forward. So we found that DECNUB-214 requires XPO1 interaction to induce the expression of these genes that I mentioned before. And when we treat the patient cells with the XPO1 inhibitors, this binding is lost, and the expression of these key leukemia genes was reduced, causing the kill of these cells, leukemia cells. So in a way, we identify a potential targeted therapy for T6-9 patients.
So that would be my question. Can you use these XPO1 inhibitors in patients? Is it known whether this is safe to use?
Yeah, that's a very interesting question, because XPO1 inhibitors, one selinexor, for example, is an XPO1 inhibitor approved for the treatment of multiple myeloma, which is great because it's approved already. So it will speed up the possibility of being used for acute myeloid leukemia treatment. However, it's very toxic. So there are new versions of XPO1 inhibitors coming now and being studied in preclinical and clinical studies. So probably in the coming years, we will have more safe XPO1 inhibitors that can definitely be tested. So I believe our research will pave the way for potential preclinical and clinical trials for the particular treatment of T69 patients.
So does this have any implication for other AML subtypes?
That's very interesting as well, and this is actually has been studied before because Hox genes are overexpressed in so many other subtypes. So in those subtypes, the role of XPO1 involved in the regulation of these genes is being currently studied by other groups. So it seems like XPO1, by the interaction through other factors, may have a role in the upregulation of these genes in other subtypes where these genes are also upregulated. So the particular use of XPO1 inhibitors is being currently studied for the treatment of other patients with the overexpression of XPO1 of Hox genes, like for example, NPM1 mutated patients or MLL rearranged patients as well.
So what are the next steps for this research?
So definitely we need to expand this research in other models before starting a clinical trial. But there are still lots of questions from the molecular perspective that are still open. So I really want to understand how these two proteins interact, through which domains, and if apart from the genes that we looked into, like Hox genes or FOXY1, as I mentioned before, if there are other key leukemia genes that are also regulated by the interaction of these two proteins, the chimeric protein and XPO1, that may have a role in leukemia, even if they've not been related to hematopoiesis or leukemia before, to identify other mechanisms that can explain this malignant transformation and how this subtype is very aggressive, as well as the interaction with FLT3-ITD mutation, because it's quite interesting that these patients with the T69 translocation also acquire the FLT3-ITD mutation in more than 70% of the cases. So it will be very interesting to see if there is any relationship between the fusion protein, FLT3-ITD, so if they have a role inside the cells, and if XPO1 is somehow involved in this. So because if that's the case, if XPO1 is somehow related with FLT3-ITD, then we can expand the use of XPO1 inhibitors for other subtypes like FLT3-ITD mutated patients, which are really, really common.
So this project was funded thanks to a grant from EHA. Can you reflect on how this grant has helped your career?
Yeah, I mean, I'm really, really grateful to the EHA, because definitely getting this grant was a key turning point on my career. So it helped me so much to my independence. I was appointed as a lecturer when I got this fellowship, because it was my first funding as a PI. So then I could establish my own research group at the Barts Cancer Institute in London. And after that, I got more funding, and I gave me the opportunity also to complete this super exciting project that we recently published. So it was a really beautiful story. Now I moved my group to Madrid today, but keeping my relationship with London. So, but everything started with the EHA research grant. So I'm really, really grateful.
So what would you tell young investigators that are thinking of applying for a young EHA grant?
Please do it. Do it, because I mean, it changed completely my career, and I think it's a great opportunity, but not just for the funding itself, that obviously is what we need to move forward in our career, for sure, but also for the visibility and because it's the prestigious of these grants is very well known in the community of hematologists and researchers in hematology. So that's one of the key things as well, because it gives you visibility. So then you get much more opportunities around, like chairing sessions, being involved, giving talks, and knowing other scientists, other hematologists. So it's like a fantastic opportunity. So please do it there. It's not only the junior one that is the one that I got, but there are more, like the advanced, and also lots of opportunities for collaboration with among different groups.
Are you involved in other ways in EHA now?
Yeah, I'm part of the mentoring program committee. So it's a new program that is launching EHA this year. So we are matching mentees and mentors, and I hope it's going to work right. Yeah, I'm part of this committee organizing this new initiative that I think is going to be fantastic, and hopefully it will continue for many years.
What is the best part of your job?
Wow. But definitively knowing people doing research in hematology. So I think it's the part that I love the most, like seeing what others do and collaborating with other people. With the project that was funded by EHA, I had the opportunity of supervising for the first time my own PhD student, and seeing how happy she was when the experiments worked, when our hypothesis, the one that I wrote when I applied for the EHA, worked as well, and it was guiding the continuation of the project. I think it was the most rewarding thing that I ever experienced. So that confirmed that is what I want to do, like research, training other people, like building my group, and see how working together we can get somewhere.
And what is more difficult in your job?
Getting funding. Getting the fund. So yeah, definitely that's the most challenging part for every researcher. I mean, the future is in the collaborations, that's for sure. So I think we can't be experts of everything. So if we want to make research that changes the way in which we treat the patients, the way in which the patients are cured and managed, we definitely need to work together with including everything, like artificial intelligence now, bioinformaticians, research and doing experiments hand-on in the lab, lots of people thinking, connecting, and talking, and doing brainstorming. So that's the future. So I think the funding will come through all these ways as well, that most of the funder bodies are also seeing that that's the way in which we should be working. So,
for clinical trials or for anything that you have to test now, so you will need a lot of collaboration and interactions.
Exactly, with industry as well. So definitely, that's the future. We can't forget the rare subtypes of acute myeloid leukemia. So because it matters, it's a really aggressive disease, but there are some subtypes within the AML that are even worse than others, and we can't forget those, because I was asked in lots of interviews why focusing on a rare subtype within a rare disease, because I mean, fortunately, AML is not really, really frequent, but it's super aggressive. Let's focus on rare subtypes as well, because they matter. They are patients. It's people behind suffering of this disease. So that's one thing, that collaborations are needed for make science bigger and to get impactful results. And that's it.
Well, thank you very much for being with us today, Dr. Río-Machín, and thank you for all the audience for listening, and stay tuned for more episodes.
Thank you very much for the invitation.